So, very good morning, good afternoon, good evening, everyone. So, today we are going to discuss first unit, that is your pathophysiology. Physiology, right? So, we are going to discuss about pathophysiology, and if you see your syllabus on pathophysiology, so you can find that first unit is about basic principles of cell injury and adaptations. So in basic principles of cell injury and adaptation, first part is introductions, definitions, homeostasis, component and types of feedback systems. So this we have already discussed in human anatomy and physiology class as well. But just a quick review, so everyone of you know that what is homeostasis. Homeostasis is, maintenance of your body environment, right? So maintenance of your body environment we called homeostasis, and this body environment is maintained through feedback systems. There is a feedback systems are of two types, positive feedback and negative feedback, right? So there are a two feedback system by which cell maintain its homeostasis. So one is your negative feedback systems, another one is your positive feedback systems, right? So in negative feedback system, the difference between these two system is negative and positive feedback system is if anything which is increase in our body, so in response to that, if body is going to decrease it, that is comes under negative feedback systems. So for examples, your blood glucose level is increase, right? Particularly it happen in the diabetes mellitus. So your blood glucose level is increased, so in response to it, your body has decreased it or maintained it. That comes under negative feedback system. But don't be confused. If something is decreased in the body, and if it's going to increase by the body, that also will be the negative feedback. That's means both will be considered as a negative feedback. So नेगेटिव फीडबैक सिस्टम का मतलब है कि कोई चीज आपकी बॉडी में बढ़ रही है, उसको बॉडी ने कम किया, या कोई चीज कम हो रही है तो बॉडी ने उसको बढ़ाया और मेंटेन किया. दैट बोथ कम्स अंडर नेगेटिव फीडबैक सिस्टम्स, राइट? पॉजिटिव फीडबैक सिस्टम मीन्स अगर आपकी बॉडी में कोई चीज बढ़ रही है, इंक्रीज हो रही है, तो बॉडी ने उसके रिस्पॉन्स में वो चीज और बढ़ाई, या बॉडी में कोई चीज कम हो रही है तो बॉडी ने उसके रिस्पॉन्स में वो चीज और कम की. दैट इज योर पॉजिटिव फीडबैक. सो समटाइम स्टूडेंट्स आर कंफ्यूज्ड कि कोई चीज कम हो रही है और बॉडी ने उसको और कम किया तो वो नेगेटिव फीडबैक है. नहीं, वो आपका पॉजिटिव फीडबैक ही है. पॉजिटिव फीडबैक डजंट मीन कोई चीज बढ़ रही है तो वो बढ़ेगी ही. कोई चीज अगर कम हो रही है बॉडी में और उसको और कम किया बॉडी ने, तो दैट इज योर पॉजिटिव फीडबैक एज वेल, right? There is a very rare examples for this, but of course that is your positive feedback. तो again I am repeating, negative feedback का मतलब है कोई चीज अगर आपकी बॉडी में बढ़ रही है, फॉर एग्जांपल आपका ब्लड शुगर बढ़ा है तो बॉडी ने उसको मेंटेन किया, कम किया, दैट इज नेगेटिव फीडबैक. फॉर एग्जांपल आपका ब्लड प्रेशर कम हुआ है, बॉडी ने उसको बढ़ाया, दैट आल्सो कम्स अंडर नेगेटिव फीडबैक. कोई चीज आपकी बॉडी में बढ़ रही है, उसके रिस्पॉन्स में बॉडी ने उसको और बढ़ाया, पॉजिटिव फीडबैक. कोई चीज आपकी बॉडी में कम हो रही है, बॉडी ने उसको और कम किया, पॉजिटिव फीडबैक, राइट? So if you remember, we have discussed classical examples for negative feedback and positive feedback. For negative feedback, if something is increased, we have discussed in detail. If your blood sugar level is increased, so how body will maintain it? So pancreatic beta cells will be stimulated. It release the insulin, and insulin, in the presence of insulin, your glucose will be uptake by your cells or tissues. So your blood glucose level is maintained. Another examples we have discussed many times. If your blood pressure is decreased, your body's baroreceptors in the blood vessels or carotid sinus sense it that your blood pressure is decreased. Signals will goes to the brain. In response to that, what the brain will do? Brain will activate the sympathetic systems. And if sympathetic system is activated, so your adrenaline, non-adrenaline, these types of neurotransmitter is released. So these adrenaline, non-adrenaline, how it increase the blood pressures? Adrenaline, non-adrenalines, they have a receptors all over the bodies. They have a beta 1 receptors on the heart. So adrenaline will stimulate the beta 1 receptors. It's a GPCR. It's going to stimulate and ultimately it increase the cardiac output. It increase the stroke volumes and stroke volumes will ultimately increase the output. And if your cardiac output is more, your blood pressures will be increased. That is the one mechanism by which your sympathetic system is increasing the blood pressure. Another mechanism is adrenaline, non-adrenaline also have a receptors on your blood vessels. There is a alpha 1 receptors present on the blood vessels. Again, these are GPCR. And when your adrenaline is going to stimulate these receptors, alpha 1 receptors in the blood vessels, your blood vessel is going to constrict and results in vasoconstrictions. So when your blood vessel is constrict, your blood pressure will be increased. Why? Because blood pressure is equal to cardiac output multiplied by total peripheral resistance. Multiplied by total peripheral resistance. So sympathetic system is increasing your blood pressures by two mechanisms. One is by stimulating the beta 1 receptors, your cardiac output is increased. By stimulating alpha 1 receptors, which are present on blood vessels, result in vasoconstriction, so increasing the total peripheral resistance. If your total peripheral resistance is increased, your blood pressure will be increased. If your cardiac output is increased, your blood pressure will be increased. So why it's happening? Because your blood pressure is decreased and body will try to maintain it. So how body is trying to maintain? First, your body should have the ability to sense that your blood pressure is decreased. So sensor has sensed it. Baroreceptor, carotid sinus are the sensor and send the signal to the brain. And in response to that, what the brain has done? So brain has stimulated your sympathetic systems, and when your sympathetic system stimulated, your adrenaline, non-adrenaline release, these adrenaline, non-adrenaline act on its receptor all over the body and increase your blood pressure. That is one of the mechanism by which body maintain your blood pressure if your blood pressure is decreased, right? Another mechanism is, this particularly when your blood pressure is decreased, so your renal blood flow is also decreased. So blood flow to the renal system is also decreased. And when renal blood flow is decreased, so there is a one hormone, well-known hormones, many times we have discussed, is released into the blood, and that hormone is your renin, right? So when renal blood flow is decreased, because if you, when your blood pressure is decreased, so your blood flow to renal system is also decreased, and in response to it, your kidney will release one hormone that is the renin. So renin, uh, you know, s- just one second. Next is renin is going to how renin is going to increase your blood pressures. So that is well-known systems that we call RAS, renin, angiotensin, aldosterone systems. When it's activated, when your blood pressure is decreased. So when your blood pressure is decreased, as I said, when your BP is decreased, so what will happen? Your renal blood flow to the renal system is also decreased. So renal blood flow is decreased, right? Renal blood flow is decreased. So when your renal blood flow is decreased, there is a release of one hormone from the kidney, and this hormone will reach where? It will reach into the systemic circulations, right? So this renin will reach in the systemic circulations. For example, this is your blood supply. So renin will release. So in plasma, there is a one protein that is synthesized by the liver. So liver is synthesized one protein. That protein name is angiotensinogen and release it in the blood circulation. So there is a protein, one protein that is name is angiotensinogen. Oh, sorry, angiotensinogen. Oh, sorry. Angio, Angiotensinogen. So this angiotensinogen plasma proteins, right? So what happens? This renin convert this angiotensinogen. This angiotensinogen is converted by the plasma. This renin convert this angiotensinogen Okay. Select. Okay. Let me check. This particularly this angiotensinogen is by the renin, this angiotensinogen is converted into angiotensin 1. This angiotensin, where these conversions are happening? These conversions are happening in the blood circulation. So this angiotensinogen is converted into angiotensin 1. And this angiotensin 1, particularly when passed through the lungs, in the lungs, there is a enzymes. That enzyme name is angiotensin converting enzymes. So that enzymes will convert, right? That enzymes will convert your, okay. So, this renin convert this angiotensinogen into angiotensin 1, right? Angiotensin 1. And this angiotensin 1, which is in your blood circulation, is converted into angiotensin 2 by the enzymes. Enzyme name is ACE, angiotensin converting enzymes. And where these conversions are happening in the blood circulation. So this angiotensin 2 have a receptors in the blood vessels. So angiotensin 2 act on the blood vessels and result in vasoconstrictions. And you know that if there is a vasoconstriction, so BP is equal to cardiac output multiplied by TPR, total peripheral resistance. So your total peripheral resistance will increase, so your blood pressure will increase. This is another mechanism by which body will increase your blood pressure. Another mechanism is this angiotensin 2 is also stimulators of your sympathetic systems. This is another mechanism, sympathetic systems. So we already discussed how sympathetic systems increase your blood pressure. So it's angiotensin 2 is a stimulator of your sympathetic systems. So adrenaline, non-adrenaline will release. So that will act on your beta 1 receptors, alpha 1 receptors on the blood vessels, beta 1 receptors on the heart. When your beta 1 receptor are stimulated, your cardiac output is increased. When your alpha 1 receptor is stimulated, which are present in the blood vessels, result in vasoconstriction. So your total peripheral resistance is increased and ultimately your blood pressure is increased. Another mechanism is this angiotensin 2 is going to stimulate your adrenal cortex, adrenal glands. There is a adrenal cortex, and from the adrenal cortex, it's going to release one hormone, and that hormone is aldosterone. That's why we call renin, angiotensins, aldosterone. So aldosterone is released by the your adrenal cortex and act on the proximal convoluted tubules of your nephron, right? So in the kidney, in the nephron, so these and aldosterone will act on the nephron and do the reabsorptions of sodium and water ion. Sodium and water is going to reabsorb. So if your sodium and water is going to reabsorb, your blood volume will increase, your cardiac output will increase, your blood pressure will increase. So these are the various mechanisms by which body is going to increase your blood pressure. So this is one of the classical examples for the negative feedback systems, right? Similarly, we have discussed positive feedback as well in detail. You can see the videos on the blood clotting. So, one of the mechanisms. Mhm. So again, if I repeat quickly how your blood pressure is increased, first blood pressure is decreased. Blood pressure is decreased, it will be sensed by your baroreceptor carotid sinus. Signals will go to the brain. In response to that, your sympathetic system is activated. Sympathetic system is going to stimulate your beta 1 receptor in the heart, cardiac output will increase, stimulate alpha 1 receptors on this blood vessels, result in vasoconstriction, total peripheral resistance will increase, and blood pressure will be increased. That is the one mechanism. Another mechanism is because blood pressure is decreased, so blood flow to the renal system is also decreased. So when blood flow to the renal system is decreased, there is a one hormone which is released into the blood circulation, that is a renin. And in the blood, there is a protein that is we called angiotensinogen. So that renin convert this angiotensinogen into angiotensin 1. And this angiotensin 1 is going to be converted into angiotensin 2 by the enzymes that is well known, we called angiotensin converting enzymes. This angiotensin 2 is a powerful stimulator of your sympathetic systems. So again, same mechanisms. Of course, the sympathetic system is stimulated here as well, but this also again, a powerful stimulator of sympathetic system. So again, it's going to be increase your blood pressure. Another mechanism is angiotensin 2 has a receptors on its blood vessels as well. So when angiotensin 2 act on the blood vessels, it results in vasoconstriction. So if blood vessel is constricted, so your total peripheral resistance is increased. Total peripheral resistance is increased, your blood pressure is increased. Another mechanism is angiotensin 2 is going to stimulate your adrenal cortex and release one hormone that is the aldosterone. And aldosterone will act on the proximal convoluted tubule and going to reabsorb sodium and water. And reabsorption of sodium and water results in increase in the blood volume and your cardiac output will be increased. And cardiac output will increase, your blood pressure will be maintained. So many times we have discussed it. Of course, so for positive feedback, classical examples are, we have already discussed their mechanism as well. For classical examples for positive feedback is, two classical example is, one is, uterine contractions during the childbirth, and another one is your blood clotting, right? In blood clotting, we have discussed in details how the blood clotting mechanisms are happening, why blood in systemic circulation doesn't clot, initially how it started, and how your primary plug is getting the strengthened by your clotting factors. So we have discussed it in details. That is another one of the classical examples of your positive feedback systems. Another one is, of course, during the childbirth, so there is a initially uterine contraction is started. There is a release of one hormone, oxytocin. Oxytocin act on its receptors in the uterine muscles. It's a GPCR. It's going to result in uterine contraction. So that mechanism also we have discussed in detail. So that is in your first unit, homeostasis, and how your homeostasis is maintained by the feedback systems. So feedback systems are two, positive feedback and negative feedback. So by these mechanisms, your homeostasis of the body is maintained. Another classical examples of the homeostasis, how it maintain is body temperature. You might have read in various textbook how body maintain the body temperatures. So body temperature is your how much? 37 degrees Celsius. Why it's 37? Because most of the enzymes optimum temperature to work is 37 degrees Celsius. So, when it's decrease or increase, so again, your homeostasis is disturbed, so body will maintain it. If your body temperature is decrease, so how body will increase it? By shivering. If your body temperature is increase, how body maintain it? By sweating. So body will decrease the temperature. So that is, we have already discussed, right, how it maintained. Now next thing is about uh, pathophysiology's introductions. When we talk about up to this, we have discussed about normal physiological process. Means we have not discussed any pathophysiological process, but it's a normal physiological process by which body maintained its homeostasis. So pathophysiology, when we talk about pathophysiology, to see this word carefully, this word is made up of two things, patho and physiology. Physiology, we have already discussed human anatomy and physiology, and I hope every one of you knows that what is physiology. Physiology is how your body works, right? And there is another word which is add up is here is patho. Physiology is how your body functions, how your body works. That is your physiology. So patho is means your disease. There are two things. Pathology. Pathology means study about the disease. Physiology means study about the functions of the body. So it's a combination of both things, patho and physiology. So how you can define pathophysiology? Pathophysiology you can define in a different way. One of the way is you can define it Pathophysiology is the branch where we study the how body functions are altered in pathological conditions. Means how our body functions. Body function is physiology. How these body functions are altered in pathological conditions. Pathological means in disease conditions. So that is pathophysiology. So what is the difference between pathophysiology? If someone asks you what is the difference between pathophysiology and pathology? Pathology. Logy means studies, right? Logy. Pathology is the study about the disease, right? Whereas pathophysiology is study about the alterations of the normal functions of the body in pathological conditions, right? So there are three things: physiology, pathology, and pathophysiology. So that is your pathophysiology, and in pathophysiology, we are going to discuss various things. So starting with the cellular adaptations. So, for example, this is your normal cell, one of the cells, normal cells. This is nucleus. So it maintains its homeostasis by the feedback systems. For example, there is any stress or stimuli, right? Any stress, noxious stimuli, anything which disturb its homeostasis. Right. Anything, any stress, any noxious stimuli, this is your normal cells. So when this normal cell is maintained its homeostasis, right? So when this homeostasis is altered by stress or altered by any noxious stimuli, so what will happen? So your cell will try to maintain or cell will try to adapt to these conditions, right? So initially, when cell is try to adapt against noxious stimuli or against any stress, so that we call cellular adaptations. So what is cellular adaptations? You can see the word cellular adaptations. Means your cell is going to adapt. And why there is a need to adapt? Adapt to adverse conditions, right? When your homeostasis is disturbed, तो cell is trying to adapt to that conditions. अगर आप कहीं पे मीन्स मान लो अभी आप कहीं बहुत ठंड में चले जाएं, पहाड़ों में चले जाए, तो योर बॉडी विल ट्राई टू अडेप्ट टू दैट कंडीशंस, राइट? अगर ये आप किसी ऐसे इन्वायरमेंट में चले जाएं जहाँ पे बहुत एडवर्स कंडीशंस है, सो योर बॉडी विल ट्राई टू अडेप्ट to that conditions. So that is your cellular adaptation, means your body is trying to adapt. This word you have to remember, trying to adapt. Adapt किस चीज के लिए करना है? जो कि आपका अगेंस्ट जो नॉक्सियस स्टिमुलाई है या स्ट्रेस है जिसने आपके होम्योस्टेसिस को बिगाड़ा है। दिस योर नॉर्मल सेल अपनी होम्योस्टेसिस कंडीशन में है, पर जब किसी स्ट्रेस ने, नॉक्सियस स्टिमुलाई ने, किसी एक्सटर्नल फैक्टर्स ने इस होम्योस्टेसिस को डिस्टर्ब किया, तो सेल विल ट्राई टू अडेप्ट टू दैट कंडीशंस, and that is we call cellular adaptations, right? So cellular adaptation, when we talk about cellular adaptation, so there are a hypertrophic, hypertrophy, hyperplasia, atrophy, there is metaplasia, there is dysplasia. So you can see that these are the mechanisms, not mechanism, types of cellular adaptations. Hypertrophy, so cell will try to adapt to these adverse conditions through these types: hyperplasia, hypertrophy, atrophy, metaplasia, and dysplasia. So these are the types of cellular adaptation. One by one we will discuss, but first you understand these are the, by these cell will try to adapt to these particular conditions. And if cell is unable, there are two things can be happen. Cell can you can adapt to that particular adverse conditions, and you will again go back and maintain its homeostasis, right? But if your stress is too much, your noxious stimuli intensity is too much, so in that conditions, you maybe your cell is unable to adapt to these conditions. So in that case, it will move from cellular adaptation to cellular injury, right? It can results in injury. जरूरी नहीं है ना कि आपकी बॉडी उस स्ट्रेस को, उस नॉक्सियस स्टिमुलाई को आपके सेल अडेप्ट कर ही ले। अगर वो बहुत ज्यादा स्ट्रेस है, बहुत ज्यादा नॉक्सियस स्टिमुलाई है, डिपेंड्स अपॉन इंडिविजुअल टू इंडिविजुअल एज वेल। कोई आदमी स्ट्रेस को अच्छे से हैंडल करता है, कोई स्ट्रेस को अच्छे से हैंडल नहीं कर पाता। तो इट डिपेंड्स अपॉन वेरियस फैक्टर्स। सो इफ योर अगर आपका सेल अडेप्ट नहीं कर पा रहा है तो इट कुड रिजल्ट इन दी इंजरी, एंड दैट वी कॉल सेलुलर इंजरी. So cellular injury is of two types. One is reversible, another one is irreversible. Cellular injury is two types, reversibles and irreversible. Reversible, as its name indicate, when you remove the stress, noxious stimuli, so again cell will go back to its normal conditions. That is a reversible injury. कि वो रिवर्स हो जाएगा, राइट? जो भी आपका, फॉर एग्जांपल जो भी आपका डैमेज हुआ वो सब रिवर्स हो जाएगा, एंड अगेन सेल विल गो बैक टू इट्स नॉर्मल कंडीशंस. इरिवर्सिबल, एज इट्स नेम इंडिकेट, इरिवर्सिबल का मतलब हुआ कि एक बार जो हो गया उसको आप दोबारा नहीं ला सकते. तो इरिवर्सिबल क्या चीज है? दैट इज डेथ. अगर कोई मर गया तो क्या आप उसको जिंदा कर पाओगे? नहीं कर पाते. तो दैट इज वी कॉल्ड सेलुलर डेथ, सेल डेथ. So irreversible cell injury means it's a cell death. DATH. Cellular death. Cell death. Cell death is cell will die through two mechanisms. One is apoptosis. 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 And another one is your necrosis. Necrosis. So there are two mechanisms by which cellular death can happen: apoptosis and necrosis, right? सो अगेन आई एम रिपीटिंग, ये आपका नॉर्मल सेल है जो कि होम्योस्टेसिस मेंटेन कर रखा है. और हमने बात कर ली कि होम्योस्टेसिस कैसे मेंटेन होता है, थ्रू पॉजिटिव फीडबैक एंड नेगेटिव फीडबैक सिस्टम्स. फिर जब भी कोई नॉक्सियस स्टिमुलाई, स्ट्रेस हुआ, तो ये होम्योस्टेसिस आपका बिगड़ गया, सेल का बिगड़ गया, तो सेल ने ट्राई किया उसको अडेप्ट करने के लिए इस कंडीशन को. दैट इज बी कॉल्ड सेलुलर अडेप्टेशन. सेलुलर अडेप्टेशन के टाइप है हाइपरट्रॉफी, हाइपरप्लेसिया, एट्रोफी, मेटाप्लेसिया, डिस्प्लेसिया. बट जरूरी नहीं है कि वो अडेप्ट कर ही ले. अगर अडेप्ट नहीं करता तो सेलुलर अडेप्टेशन मूव किस साइड हो जाएगा? दैट विल रिजल्ट इन सेलुलर इंजरी. सो सेलुलर इंजरी इज ऑफ टू टाइप्स, रिवर्सिबल एंड इरिवर्सिबल. रिवर्सिबल का मतलब है कि आपने नॉक्सियस स्टिमुलाई और स्ट्रेस अगर रिलीज किया तो वो बैक टू नॉर्मल स्टेट में आ जाएगा. अगर आपने नहीं किया, मीन्स वो इंजरी इरिवर्सिबल भी हो सकती है, जिसको हम सेल डेथ बोलते हैं. दैट इज योर एपोप्टोसिस एंड नेक्रोसिस, right? I hope it's clear. Now, one by one, we are going to discuss about cellular adaptations, right? So, first one is your hypertrophy, right? Mhm. If you have any questions, you can put in the chat box as well. Hypertrophy. First first types of cellular adaptation which we are going to study is hypertrophy. So, what is hypertrophy? Hyper means what? Hyper means increase in size, right? Sorry, hyper means increase, trophy means size. So increase in the size of the cells we called hypertrophy. Got it? Just 1 second. Yeah, sorry. So hyper means increase. Hyper का मतलब क्या होता है? Increase. Trophy का मतलब होता है size. So increase in the size of the cells. If you have to be present it in a diagrammatical way, so आप एक सेल बना दीजिए, राइट? आफ्टर ट्रॉफी, हाइपरट्रॉफी में आपका इसका साइज बड़ा करके दिखा दीजिए. दिस इज द हाइपरट्रॉफी, इंक्रीज इन द साइज ऑफ द सेल्स, राइट? अगर हाइपरप्लेसिया की बात करें, हाइपरप्लेसिया, हाइपर मीन्स again increase, but plasia means number. So increase in the number of the cells. So how you can present, this is your cells, तो एक से आपके फॉर एग्जाम्पल 4 सेल बन गए, तो दिस इज योर hyperplasia, increase in the number of the cells, right? Hypertrophy, hyperplasia. So particularly hypertrophy you can see particularly जब आप फॉर एग्जाम्पल आपका मसल मास इंक्रीज हुआ, राइट? आप जिम गए, 1 महीना जिम जा रहे हो, तो आपकी बॉडी बिल्डिंग हो रही है, तो दैट इज योर इंक्रीज इन द साइज ऑफ द मसल्स, राइट? सो दैट इज योर मस्कुलर हाइपरट्रॉफी, तो आपका साइज इंक्रीज हो गया सेल्स का, राइट? व्हाट इज स्ट्रेस इन दिस कंडीशन? स्ट्रेस इज योर एक्सरसाइज, आप डेली एक्सरसाइज कर रहे हो, जिम जा रहे हो, तो दैट इज अ स्ट्रेस। अब वो स्ट्रेस को अडेप्ट करने के लिए बॉडी ने क्या किया, अपने मसल्स का साइज बढ़ा लिया, सिंपल, राइट? स्ट्रेस इज योर एक्सरसाइज, आप एक्सरसाइज मीन्स जो आप जिम में हैवी एक्सरसाइज कर रहे हो, दैट इज अ स्ट्रेस, तो बॉडी ने उसको अडेप्ट करने के लिए मसल्स की हाइपरट्रॉफी कर ली, राइट? There is a various, so examples of hypertrophies. Your ventricles, when your ventricles are under stress for a longer period of time, so their ventricular hypertrophy happens. So your ventricles, hypertrophy, increase in the size of the cells, myocardial cells will happens in that case. So another thing is your increase in the size of the breast during the pregnancies, during the puberties. So that is hypertrophy as well as hyperplasia. But hyperplasia is more in that case as compared to hypertrophies. So there are a various example for the hypertrophy. But most important thing is what is the mechanism of hypertrophy, how cells increase its size. So to increase the size, you need more proteins, right? तभी अगर आप उसमें जाते हो, जिम में जाते हो, तो वो ट्रेनर क्या बोलता है, भाई प्रोटीन डाइट आपको ध्यान देनी पड़ेगी, मीन्स आपकी एक्सरसाइज के साथ आपकी डाइट प्लान भी चलेगा, राइट? तो उसमें प्रोटीन्स होते हैं, तो इंक्रीज इन प्रोटीन सिंथेसिस विच इज रिक्वायर्ड फॉर द मसल्स, राइट? To increase the size of the cells. So how it increase? So there are various mechanisms by which your increase in the size of the cells is happening. One of the well-known mechanism is your signaling pathways which promote the muscle growth is your IGF-1. IGF-1, one of the pathway which increase or promote muscle growth is IGF-1, PI3K, PI3K, right? Then there is a AKT, AKT, then there is a mTOR. This is the one of the pathways which promote muscle growth. IGF-1, PI3K, AKT, mTOR pathway. So why we are writing like this? क्योंकि सबसे पहले ये, फिर इसके बाद ये एक्टिवेट होगा, फिर इसके बाद ये, इसके बाद ये, एंड देन इट रिजल्ट्स इन इंक्रीज इन दी protein synthesis. So IGF-1 is insulin growth factor. What is IGF-1 is insulin growth factors, and it's a potent anabolic factors, right? It's produced by the livers. So IGF-1, as I said, starting molecule कौन सा है, IGF-1, उसके बाद ये सीरीज में एक्टिवेट हो रहा है। तो IGF-1 which is produced by the liver, it act on its receptors. इसका रिसेप्टर्स का नाम, थैंक गॉड इट्स सिंपल नेम, इसका नाम IGF-1 रिसेप्टर ही नाम है, IGF-1 रिसेप्टर्स। तो ये मॉलिक्यूल अपने रिसेप्टर्स पे बाइंड होगा, और अगर आपको ध्यान होगा, मैंने कई बार आप लोगों को बताया है, रिसेप्टर्स हमारे 4 कैटेगरी के हैं। वन इज योर GPCR, फैमिली की बात करें, GPCR, जी प्रोटीन कपल्ड रिसेप्टर्स। एक हमारी फैमिली है इंजाइम लिंक्ड काइनेज रिसेप्टर की, एंजाइम लिंक्ड काइनेज रिसेप्टर्स, एक हमारी है लिगेंड गेटेड आयन चैनल रिसेप्टर्स, और एक हमारी है न्यूक्लियर रिसेप्टर्स। तो जब हम बात कर रहे हैं IGF-1R, तो नेक्स्ट क्वेश्चन शुड अराइज इन योर माइंड इज कौन सी फैमिली का रिसेप्टर्स है ये? या तो GPCR होगा, या एंजाइम लिंक्ड काइनेज होगा, या लिगेंड गेटेड होगा, या न्यूक्लियर होगा। तो इस केस में दिस वन इज योर एंजाइम लिंक्ड काइनेज रिसेप्टर। तो IGF-1R इज एंजाइम लिंक्ड काइनेज रिसेप्टर्स। जब हमने बात की एंजाइम लिंक्ड काइनेज रिसेप्टर, तो आपके ब्रेन में एक मैप बन जाना चाहिए। एंजाइम लिंक्ड, व्हाट इज द कॉन्फ़िगरेशंस ऑफ योर एंजाइम लिंक्ड काइनेज रिसेप्टर? सो व्हाट इज कॉन्फ़िगरेशंस? इट्स ए सेल मेम्ब्रेन रिसेप्टर्स, right? So it says cell membrane. One part is outside the cells that we called extracellular domain. Another part is inside the cell membrane that we called intracellular domain. सेल के बाहर वाली डोमेन को एक्स्ट्रा सेलुलर डोमेन, सेल के अंदर वाली डोमेन को इंट्रासेलुलर डोमेन. सो दिस receptors normally presents in the monomer form. ऐसे ये अलग अलग रहती हैं यूनिट, जब when there is no ligand bind with the active sites. So I told you, कोई भी मॉलिक्यूल जो हमारे काम करते हैं, they are not going to bind with whole receptors. They are going to bind with particular site, और उस site को हम बोलते हैं active site. So when there is no ligand bind in the active site of the receptors, this present in the monomer form. Monomer, right? Monomer. When your extracellular ligand is going to bind, and extracellular ligand in this case is, what is the name of extracellular ligand in this case is IGF-1, insulin growth factors 1. This is your ligand which is going to bind with IGF-1 receptors, and IGF-1 receptor is enzyme linked kinase receptor. So it's going to bind. When it's going to bind with the extracellular domain, what will happen? These two units is going to attach each others. So inactive state of enzyme linked receptor is monomer, but when your ligand bind with the active site, this monomer will convert into dimer. Now process is going to start. Got it? So it becomes dimer. So when this is the active configurations, if question is asked, what is the active configurations of your enzyme linked receptors, and options are monomer, dimers, trimers, whatever. So answer to this question was dimer, right? So when it's activated, now in the intracellular domain is this one inside the cells. This have a, because receptors are made up of proteins. Proteins are made up of amino acids. So it results in the phosphorylations of tyrosine residues here. Phosphorylations of that will happens, right? When phosphorylation is happens, so there is a insulin receptor substrate, IRS. So this IRS is going to bind, because after phosphorylations of tyrosines amino acids, it creates the active sites or docking sites for the insulin receptor substrate. So your insulin receptor substrate is going to bind with that and going to activate. Next molecule is, now you can see, when you remember this pathway, first molecule was IGF-1, which bind. Now next molecule which is going to activate is PI3K. So when your IRS bind, it's going to activate PI3K. PI3K is phosphoinositol 3 kinase, right? Phosphoinositol 3 kinase. So this phosphoinositol 3 kinase is going to do the phosphorylations of PIP2 into PIP3. इट गोइंग टू फॉस्फोराइलेट. फॉस्फोराइलेशन का मतलब है एक फॉस्फेट ग्रुप ऐड हो गया. तो पहले तो फॉस्फोइनोसिटोल डाइफॉस्फेट, फॉस्फोइनोसिटोल डाइफॉस्फेट इज कन्वर्टेड टू फॉस्फोइनोसिटोल ट्राइफॉस्फेट, right? Now you can see this is your second. Now next molecule is AKT. So which molecule is going to activate? AKT. AKT is also known as phosphokinase B. AKT, right? Now AKT is further going to, AKT has various targets. One of the target is it's going to activate your mTOR. mTOR is mammalian target of rapamycin. Mammalian target of rapamycin, mTOR, right? And mTOR further is going to make the complex with mTOR C1. And mTOR C1 is particularly, responsible for protein synthesis, mitochondrial biogenesis, and so on. And by that one, your cell size is going to increase. This is the mechanism. Got it? For clarity, I am going to explain you again step by step. See, first one is insulin growth factor, IGF-1, right? Insulin growth factor. It's going to bind with its receptors. Receptor is enzyme linked kinase receptors. So when it's bind with the receptors, your receptor is going to convert into dimer, right? Dimer is the active state. Then it results in the phosphorylations of tyrosine residues. Tyrosine residues are in intracellular domain. So it's going to, IRS is going to bind with its site, that is your insulin receptor substrate. Then this IRS is going to activate PI3K, phosphoinositol 3 kinases. Phosphoinositol 3 kinases is going to do the phosphorylation of PIP2 into PIP3, right? PIP3. PIP3 is going to activate your AKT, phosphokinase B. AKT is going to activate your mTOR, mammalian target of rapamycin. This is going to, making the complex mTOR C1. There is a C2 as well, but C1 has a role in hypertrophy, C1. So this C1 is going to do the protein synthesis, right? So increase the protein synthesis to increase the size of the cell. This is the mechanism of your hypertrophy or hyperplasia, because protein synthesis is required for hyperplasia as well. But I will show you the difference in the mechanism of hypertrophy and hyperplasia as well. This is one mechanism. Got it? Right. So next is this AKT. If you remember, I told you it's a negative regulator of FOXO. FOXO. FOXO is fork head box O. Fork head. Fork FORK. Fork. Fork head O. Fork head O. Right. So this, FOXO is act as a negatively regulator of hypertrophy. As I said, when this pathway is activated, this protein synthesis is happening. So that means that is helping in the hypertrophy. But this AKT, when AKT is activated, it's going to inhibit the FOXO. Why? Because FOXO is the negative regulator of hypertrophy. Because what the FOXO do? FOXO induce expression of gene further. It act on the nucleus, and do the expression of such kind of gene which will do the protein degradations. Protein degradations. Protein will be degraded when FOXO is activated. So in this case, what mechanism we are discussing? We are discussing the mechanism of hypertrophy where we want to synthesize a protein, right? अगर ये एक्टिवेट होगा, ये एक्टिवेट होता है, तो ये तो प्रोटीन को डिग्रेड करेगा. तो दैट्स व्हाई AKT इज इनहिबिटिंग द फॉक्सो. तो प्रोटीन डिग्रेडेशन कम होगी, प्रोटीन डिग्रेडेशन कम होगी तो आपका हेल्प करेगा हाइपरट्रॉफी में, राइट? So you have to remember FOXO is the negative regulator of hypertrophy, right? Then there is another particular after AKT, there is a GSK3 beta. GSK3 beta, glycogen synthase kinase 3 beta. So this glycogen synthase kinase 3 beta is a active serine threokinase kinases, and it's a negative regulators of cell growth. So how it happens. It's little bit different from the FOXO, because when AKT is activated, AKT phosphorylate for GSK3 beta. So AKT, what AKT do? AKT do the phosphorylation of GSK3 beta, and after phosphorylations, its function is inhibited. It's again is a negative regulator of hypertrophy, but slightly different. AKT is going to phosphorylate GSK3 beta, and phosphorylation of GSK3 beta is further going to inhibit. After phosphorylation, its function is going to be inhibited. So in summary, if someone asks you, GSK3 beta is again negative regulator of hypertrophy. अगर फॉक्सो और GSK3 बीटा बढ़ता है तो अल्टीमेटली योर प्रोटीन डिग्रेडेशन विल हैपन, राइट? So that is your mechanism of your particular hypertrophy. And when we talk about hyperplasia, so there is a CDK, cyclin dependent kinases, which will propel your your labile cells or stationary cells into the cell cycle phase. So G0 to G1, then if you remember, we talk about then synthesis phase, then G2 phase, then there is a mitosis phase, and cell will divide. So so these AKT, mTOR, these are the regulators of your cell differentiation as well, right? So these particular pathways. One more pathway is there, that is the RAS, RAF, MAPKC, ERK pathway as well, which also helps in the protein synthesis, right? So that is also similar mechanisms, means that is also showing the action through enzyme-linked kinase receptors. So if I tell you a little bit about that pathway is, pathway name is RAS, RAS, then RAF, RAS, RAF, then there is a MAPK, MAPK, then there is a ERK. So complete name of this pathway is this: RAS, RAF, MAP, ERK pathway. So again, starting point is your RAS molecule, right? So the RAS, your your growth factors is going to act on your enzyme-linked kinase receptors again, and in again enzyme-linked kinase receptor is in monomer form. Then it will become dimer. Then it's going to activate RAS. Then RAS is going to activate RAF. RAF is going to activate MAP. MAP is going to activate ERK. This ERK is going to act on the nucleus, right? And do the transcription and translation process. So like your CGen, CFOS, like these genes is going to activate CFOS, which are helps in the protein synthesis, cell differentiation, so cell growth, it helps. So this is another mechanisms when we talk about hypertrophy, right? Or hyperplasia as well, right? Any questions, anything? So hypertrophy and hyperplasia, as I said, when we talk about hypertrophies, hyperplasia, there is a increase in the protein synthesis. Now, next one is atrophy. Atrophy means shrinkage of the cells. Atrophy. So what is atrophy? Atrophy means this is your normal cells, right? So when this normal cell is going to be shrink, छोटा हो गया, that is atrophy. So, in atrophy, so your FOXO should be activated or FOXO should be inhibited. FOXO is the negative regulator or positive regulator of atrophy. It's a positive regulator, right? GSK3 beta is the positive regulator or negative regulator of atrophy. It's a Positive regulators. AKT is negative regulators or positive regulators? It's a negative regulators, right? So, sir, in atrophy, what happens? There is another well-known pathway is ubiquitin proteasome pathway. Let me type the name. ubiquitin proteasome pathway. This one, ubiquitin proteasome pathways. So for simpler understandings, and particularly atrophy is happening when there is a muscle wasting, जब आप मीन्स अपने मसल्स का यूज नहीं कर रहे हो फॉर ए प्रोलॉन्ग पीरियड ऑफ टाइम, सो इट रिजल्ट्स इन एट्रोफी. आप फास्टिंग बहुत ज्यादा कर रहे हो, 20 दिन हो गए फास्टिंग किए हुए, 30 दिन हो गए, तो एट्रोफी विल हैपन्स, राइट? Atrophy will happens. So how atrophy will happens? So when you are doing fastings or so you are doing nutritional deficiencies there. So your PI3K AKT signaling pathways are going to suppress. We have discussed about PI3K AKT mTOR pathway. So it's going to suppress. You know that if AKT is going to suppress, right? What will happens? It's going to be do the dephosphorylations, right? Dephosphorylations of GSK3 beta, because when it's activated, so there is a phosphorylation of GSK3 beta, right? But now it's dephosphorylated, now it's going to activate, right? And it results in protein degradations. Similarly, it's going to activate when it's suppressed. So there is activation of FOXO, like fork head. We have already discussed. FOXO means fork head box O. So it act on nucleus and do the transcription translation process. And during this transcription translation process, it's going to produce some enzymes which will do the protein degradation. So your protein is going to degrade. If your protein is going to degrade, it results in atrophy, right? So, particularly there is a, so why it's a ubiquitin proteasome pathway. So this ubiquitin is a small protein which covalently attached with the target proteins. For example, this is the proteins, right? Now you want to degrade this protein because this atrophy is going to happen. So protein should be degraded. So how it will be degraded? So ubiquitin proteins will be synthesized during the transcription translation process, which is maybe activated by your FOXO. So that protein is going to, that protein name is ubiquitin. So this ubiquitin is going to attach with this one, this protein, right? Attach with the target proteins, and when it attach with the target proteins, proteasome will identify it, proteasome, and do the degradation of this protein. I hope it's clear. Again, I am repeating. Ubiquitin is a small proteins which covalently attach to the target proteins. When it attaches with these target proteins, proteasome will identify it and do the degradations. That's it. And from where ubiquitins you are getting ubiquitin, behind ubiquitin, these are the signaling pathway which are producing the ubiquitin. Ubiquitin is attaching with the target protein and doing the protein degradations. So that is your atrophy, right? Protein degradations and shrinkage of your cell. This is the another mechanisms. Now there is a metaplasia is another cellular adaptations. Metaplasia means changing of one cell type to another cell type, right? When there is a prolonged stress, prolonged exposure, so how cell will try to adapt it. So, when we talk about GIT tract, right? So in esophagus, the cells are your squamous epithelium is there, right? Squamous means flat cells are there. In intestines or stomachs, there are columnar cells, right? columnar epithelium is there, and you know that columnar what it is, and why nature has done like this, because columnar cells are more resistant to, HCLs, particularly hydrochloric acid, which you are getting in the GIT tract. This is normal physiological condition. So there is a one disease that we call GERD, gastrointestinal gastroesophageal reflux disease. So in gastroesophageal reflux disease, what happens in some patients? What will happen? This particular gastric content is returning back towards the esophagus. तो यहाँ ऊपर की तरफ जाएगा. When this gastric content is going towards the esophagus, so what will happen? It's going to damage your squamous epitheliums. And as I said, columnar epithelium is more protective as compared to squamous epithelium. So what happened? These squamous epithelium is going to convert into columnar epithelium, and that is the one of the example of metaplasia. So what will happen? For example, this is your squamous epithelium where flat cells are arranged. So in gastroesophageal reflux disease, what will happen? These cells is going to convert into a columnar type. There is a formation of goblet cells are also there, which will do the production of mucus as well. So this is the one of the classical examples of the metaplasia. We also called it Barrett esophagus, right? Barrett esophagus, right? Another classical example for metaplasia is, your smoking. So in the respiratory tract, which type of epithelial is there in the respiratory tract? Respiratory tract epithelia is your columnar epithelium is there, right, in respiratory tract. So when you are doing a smoking for a longer period of time, so what will happen? These columnar cells is going to change into the squamous epithelium, right? That is also one of the classical example of metaplasia where one type of cell is converted into another cell type. It happens in the smokers. Got it? So this is about your cellular adaptations. When we talk about dysplasia is your when your cells are changing into the abnormal size or shape. That is we call dysplasia. Abnormal size and shape, right? And these could results into the cancer as well, or neoplasia as well, right? So this is about your cellular adaptations, right? How cell try to adapt and what are their possible mechanisms, right? So what we have learned so far, I told you cell is maintaining the homeostasis through feedback system, positive and negative feedback system. But when cell homeostasis is disturbed by stress or anything, then what will happen? Cell will try to adapt by hypertrophy, hyperplasia, metaplasia, atrophy, dysplasia. But if cell is still unable to adapt, then it could result in the cellular injury. A cellular injury is of two types, reversible and irreversible. And reversible injury means when you remove the stress, again it will return back to its normal state. Irreversible means once it's lost, it cannot be returned back. That is cell death, that is of apoptosis or necrosis, right? Apoptosis and necrosis. So then we have discussed one by one: hypertrophy, hyperplasia, metaplasia. So hypertrophy increase in the size of the cells, but important thing is about its mechanism. One of another mechanism which I forget to tell is about JAK-STAT signaling pathway is also there, which helps in the cell survival, cell differentiations, right? JAK-STAT pathway. JAK is Janus kinases. STAT is signal transducer activator of transcriptions. So again, it's a JAK-STAT. So first JAK is going to activate the STAT, right? And beauty is all these receptors are your enzyme linked kinase receptors, belongs to the family of enzyme linked kinase receptor. So if they belongs to enzyme linked kinase receptors, so same configurations you have to draw. Extracellular domains, there is a monomers, then dimers. So in this case, in the intracellular domains, the molecule is your JAK, Janus kinases. And that Janus kinase is going to activate your STAT. And STAT, as its name indicates, it's a signal transducer and activator of transcription. So transcription is happening where? In the nucleus. So the STAT is act on the nucleus and then do the synthesis and release of various cytokines or proteins as well, which helps in the cell differentiations, cell survivals or proteins. Right. So this is all about your cellular adaptations. So next we will discuss in the next videos about the causes of your cell injuries. If we talk about what are the reason for cell injury, so this can be physicals, means mechanical trauma, chemicals, and biological. So we will discuss in details what are the different physicals agents which results in cellular injury, chemical agents, and biological agents. There are three types of agents which could result in the cell injury, right? So this is all about your what we have covered so far. So I hope all the concepts is clear in your mind. If still you have any doubt, any questions, you can put in the comment box. I will try to answer these questions or your queries. Thank you.